Biology, Cambridge. For now, I will just stop. So you can just Google to know more about him, because if I continue, it will run out of time. I want everyone here to ensure that you are getting the gist from one of the world's best. And there's also a session definitely for questions and answers. So as I've been inviting him to speak, I want you to like drop whatever questions you have in the chat box and we'll get to them. So without further ado, I want to invite Dr. Madan to take the stage. Welcome, sir. Thank you. We are very happy to have you here.
Thank you, Talani. Thank you, Michael. Now, few things here. Thank you so much for this invitation to be with all of you today. Thank you for the brief introduction. It's always a delight to be with people who are starting on this journey to doing exciting things. I was very fortunate, 1982, to get a scholarship to come to Cambridge. Now, the story goes like this. There is a connection with Nigeria, and I feel additionally happy to share this story with you. In 1982, I completed my master's in genetics and plant breeding in India. You're right, Tulani. I studied in Haryana, the north of India, about 150 kilometers northwest of New Delhi. And I wanted to come to Cambridge. It's always been a dream since a school child to come to Cambridge, because this is the home of DNA. This is where the structure of DNA was discovered in 1953 by Watson Crick. So it was a big dream that I wanted to come to this city and spend time and learn about DNA, the magical molecule. And in 1982, this happened. In 81, I was interviewed and I managed to get a scholarship from an organization called the INLAX Foundation. Now, the story that I want to present in the connection with Nigeria goes like this. The person who set up this foundation, INLAX Foundation, set it up with the sole purpose of sending students from India to Cambridge and Oxford and some of the best schools, best universities around the world to go and learn, do something good. Now, he studied here in Cambridge. He was a student at Clare College, one of the 31 colleges here in the small town. And his father also studied here, was a senior administrative officer from India. And he studied in St. John's, again, one of the other colleges here. Now, he studied here, he completed his degree, and then what did he do? The most amazing thing, he went to Nigeria. Mr. Lakshmi, no, Mr. Shivdasani is the name, and he went to Nigeria. And he set up an industry in Nigeria, processing food. And he made a lot of money. And he took some of that money that he made and he said, I'm going to make a foundation so that students can go and follow the path that he and his father did. And if you Google right now, INLAX in Lagos, where you are seated today, you will see a hospital there called the INLAX Hospital. For those of you in Lagos, you may know this hospital. So I came to Cambridge because a foundation was set up by a gentleman who came to Nigeria and made a lot of money in Nigeria, and they funded me to come to Cambridge. Isn't that, I find this quite amazing that it's come full circle, and I'm so happy to be connecting with Nigeria. So it seems like I'm doing my duty in reciprocating what Nigeria did for me indirectly. So thanks to all of you there for drawing me into this. Sometimes you think, here we are. I don't know what that thing called God is how we So thanks to all of you for connecting me and connecting with me, and maybe even towards Nigeria. I don't know what future hope. Maybe I turn up in Nigeria. I don't know. That's the story to get started. Now, fortunate to be here. The year after I came to this, to Cambridge, was the third discovery and the publication of this structure 3, and I was so still there, alive, relatively kind of youngish, I would say. So I could meet Watson and Crick and Sidney Brenner and all the other people involved in what the big story that has grown out into biotechnology and molecular biology and what we, all the kind of things we are doing today. And I was fortunate to be in this city, to meet with these people. They all seem like heroes of the past because many of them have passed on, and they've left their contributions in big ways. So what I would like, and next year is the 70th anniversary of the discovery of the structure of DNA. So I'm fortunate to be here again. I don't know what celebrations are planned, but many of the people who were involved in all that work are no more. And I feel privileged that I can take this opportunity to form a link between that past that I was connected with and was engaged with, and I had the good fortune to meet with people who are doing this, and with the next generation. All of you are the next generation, I guess, who are going to be working on the foundations that were set up by the great minds in the city, and then to go on. And one of those things that came out of all this work is bioinformatics. So as part of this organization, Biohack, and based on what I've discussed with Michael and others, I felt that it'd be useful if we just chatted about take you through what is in my mind. It's going to be a journey through where technology is become so important, and it's going to get even more important. Within that is whole activity of a very small area that didn't exist at that time. And I just want to take you initially through one or two slides about how this whole area came about, the history from 1980 onwards, and the few key people, again, based in this town, who then helped set up what grew and has grown into what we know today. So my job, partly number one, to connect with all of you in Nigeria, to say thank you for helping Mr. Shiv Dasani come to Nigeria and make all the money that he did there, and set up a foundation, and then gave me a scholarship, and then to bring me back to where the story begins. And in that similar historic way, I just want to take you people, hold your hands and take you to where it started and where we are today and where it needs to go, and where the title of this day's discussion about how some understanding of that history and what bioinformatics is and where it's going, how this might give you an advantage when it comes to taking on your journey of going to do a master's and a PhD and setting yourself up for the rest of your life. So that's the journey that we're going to do. Now, I had a chat with Michael earlier today, and we discussed the format for this presentation. And I said, I don't want this to be a one-sided thing where I'm just putting stuff onto you. So I'd rather have an opportunity to turn into some kind of vibrant discussion as we go along, so that you'd have questions and I can point you to things, having initially taken you through some of that information. So, Tolani, that is what's in my mind, and I hope that's so. But I can guarantee you that we will see some very interesting things that will certainly surprise you, and that might even surprise me as we go through this talk, because I'm going to take you through a few slides I prepared, partly to take you through to that initial history. We talked about 1953 was a special time. It seems like a long time ago. 53, I don't know, if it's still around, but it's not too far. It's easy to connect. It's not like what the other area of biology that I work with, which is ancient systems of medicine. The oldest textbook of medicine, extant textbook of medicine, is a book called Charaka Samhita, which was the most recent redaction of this textbook. It is a textbook. It's not a reference document. It's a textbook. Is about 2,500 years old, maybe 3,000 years. The most recent redaction, there were earlier versions of this text. So that's a long time back in history compared to 30 years or 60 years or 70 years ago. So I will take you through the short journey, and then if somebody's interested, take you into the bigger, longer journey of 5,000 years and knowledge for 5,000 years, and how with bioinformatics, and only with the help of bioinformatics, will we be able to pierce our way back in time and connect with knowledge from 5,000 years ago that is ironically guiding us to the future where biology wants to go. So shall we begin? I'm going to see if I can share my slides. Now, Tolani, what is going to happen here?
Of course you can.
Slide.
Yeah, you
okay.
can. Yeah.
Okay, Wale, thank you. I'm going to share. Tell me if you can see my slides. Okay, if there's a problem, then we correct it and we see how to get it. But we've tested it with Michael. We tested it before. Now, who is managing this? I'm letting people into the room, or is somebody there? Can you see messages of people who are outside and want to enter the room, or you can see it too? Okay, so I'm quicker than both of you in letting people in. Okay, so I'm not going to do that, because I'm going to try and focus on other problems here, and let us start now. Is my screen visible?
Yes, it is.
Okay, I'm going to go to full screen now. Here we are. Is that visible?
Yes, it is.
Okay, and I'm going to turn my camera off if I can, so that I can get rid of this stuff. I think right now on the top right-hand corner, there might be my camera showing there, Tolani. I need to turn that off. And to turn that off, I'm going to click the video off, and there we are. That should turn me off. So it should look quite clear there on the screen. Thank you so much for, again, having me here. For those who want to contact me and reach me, they can take a screenshot of this. This has my contact details. If you're not able to make screenshots based on the device you're using, then don't worry. Tolani has details, or Michael has details, and they'll be able to share. So that's me. I'm based here. I came in 1982 to Cambridge, so it's my 40th year here in Cambridge, and it feels like 40 years have gone by just like that. And year seems just a very small in terms of time. I still feel like I'm 23, but that's a different matter. Now, I've started with this one slide, one image here, and this comes from real technology. And you can access this online at that link below. And I just want to read, would like to just read the first paragraph of this page. The title of that section is Computers, Databases, and Automation. And it says the introduction of computers. I'm just going to read this. I know it's on the screen, but I just want to read this to touch a few points so that we can just extend them and discuss a little more. It says much of the sequencing of the human viruses could not have been done without the aid of computers. In the very early days, most of the sequences were copied out by hand, with different workers recording particular portions of the molecule they were working on in their own notebooks. Writing them out by hand, however, became increasingly prone to error as the sequences grew longer. Amalgamating the data from the different notebooks was also very laborious. So, some idea of the scale of the effort can be seen in the first genome that was ever sequenced. So 5X174 is a bacteriophage. And the sequencing was done here in Cambridge. So this is the first genome to be sequenced. Yes, it was viral genome. And you can see, as it says, there were 9 different workers involved in recording the original data. And if you access that website, the reference, Hutchison, 2007, etc., so many other things. And shown on the left-hand side is Mike Smith. And it says Smith was a British biochemist based at the University of British Columbia in Vancouver, who is at LMB, the Lab of Molecular Biology. LMB stands for Lab of Molecular Biology. In the 1970s, in 1983, he shared the Nobel Prize with Carrie Mullis for co-developing site-directed mutagenesis. For those who know, Carrie Mullis is the man who developed polymerase chain reaction. He's no more, but he was quite an amazing person who gave us one of the most important techniques in the research. To introduce you to computers and computer programming and how that came into DNA sequence that was developed by Fred Sanger, again here in Cambridge. Fred Sanger was a very special person because he's one of the 3 double Nobel Prize winners in the world. The 2 others being the lady Marie Curie, the lady who discovered radioactivity, John Bardeen, the person who discovered some semiconductors and transistors and all of that. That's controlling technology of the kind that we're discussing and using today. And Fred Sanger, who discovered a way to sequence proteins. And about 25 years later, having won a Nobel Prize for that, went on to win the Nobel Prize for showing us how to sequence nucleic acids, RNA and DNA. And as you saw from previous paragraph, there was a problem. There was sequencing, and there was sequence information that was being written down by hand, and then to put them together, to assemble them, you needed a program. And shown on the left here is young Roger Staden. I don't know how old Roger is today. It might be 70 plus, I guess. And Roger devised something that is today recognized, almost historic, but still being used, the Staden package. Roger devised DNA sequencing software. And I'll read again from what's on the screen. His programming software would go on to be acknowledged internationally as one of the best for use for managing data from sequencing projects. Now, all of this work was driven and sponsored by the United Kingdom's Medical Research Council. So what you can see is that emphasis on medical research as being a driver, not only for doing medical research, but also for developing amazing technologies. And Roger is very special. He lives very close to my house. I live very close to the lab, about less than 300 meters from the lab. And we are all people who kind of hang around the old lab of molecular biology. The lab has since grown, and there's a new building. If you go to the website, you can access it online, Medical Research Council Laboratory of Molecular Biology. Then you can see all the work that's going on there. I'll come back to the website of the LMB just to run you through the historic account of what is archived in the Lab of Molecular Biology. Now, Roger Staden went on to write a very interesting paper called The Computer Program to enter DNA gel reading data into a computer. So remember, when we go into the history, at one time, people were writing things by hand. So you had radioactivity, you had autoradiographs, and then you grab data from autoradiographs, and then you had to read the sequence, and using the data that is generated using the Fred Sanger's dideoxy sequencing method, and then you had to write it by hand. And then Roger came along and said, well, why don't we record things directly from the autoradiograph? In fact, if you go to that link, you will see a very good historic account with a lot of figures about that whole process. So I suggest you look at that What is Biotechnology link. Again, I can share that link on the chat box for those who cannot access it. So Roger then went on to write this small paper, and this paper is very interesting because the abstract is just one line. The paper shown in purple there, it reads like this. This paper describes a simple program that uses a digitizing device into a computer. Tolami, I just want to stop here just to ask if my internet, is my sound clear? If it is not, please tell me.
Okay, I think it's clear on my end. How about you, Wale?
It's clear. Okay.
Yeah, averagely it's clear.
It's clear. Thank you. So what is fascinating, if you count the number of letters in this, I think this might be one of the shortest abstracts ever written for a research paper, for such an important research paper. I don't know how many words in here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21 words, one line. And I wish more people would write papers like this, short abstracts, punchy papers that change the history and scope of research. There's an old saying that research marches on the feet of methodology. And the methodology that Fred Sanger gave us and the different kinds of techniques he offered us advanced science, advanced biology, advanced our understanding of DNA. And it didn't stop there. The lab continued to work, and Roger Staden came along and he gave us more methodology that helped progress things. Now, so that was 1984 when he published, but clearly all the coding that he was doing, I don't know what language he was using, maybe, I don't think C existed then. I think they were writing in assembler or something. But all that knowledge led on to where we are today. And this is where we are today. And I've shown here the abstract of another paper. This is a paper that comes from New York, and it's about taste and taste receptors, how you taste different things and how that message is conveyed from what you taste to the tongue and onward to the brain. And if you look at the bit that I've highlighted here, which is single cell RNA sequencing on 800 neurons from the geniculate ganglion. I want to stop here for a second and point out why I have picked this paper, because taste in the Indian systems I earlier mentioned, there was an old textbook of medicine which dates back to the most recent redaction about 2,500 years ago. That textbook talks about taste as a therapeutic intervention. It talks about 6 tastes, and it says describes how the different tastes can be used to manage disease, to manage health. And I have a feeling that this is the future of medicine. And hence I picked up this single paper. Now, the other thing I want to point out are those cycles within cycles. RNA sequencing is what Fred Sanger gave and did his first Nobel Prize. We've come full circle from when he won his first Nobel Prize and his second Nobel Prize to where we are today. That is a long time afterwards. We seem to have come back many decades on. We're back to RNA sequencing. And we accept that we have improved the resolution of our studies and the sample size that we are using. So we are reducing sample size from whole organisms to tissues, organs, and crushing the liver and extracting DNA and RNA and studying. From all of that, we've reached a point of resolution where we are taking single cells from a given part of the brain or part of the nervous system, and then analyzing those things at very high resolution to understand processes like how tastes are sensed by the tongue and how those messages go to different parts of the body, and pointing us to the accessibility of deep biology. And as I said, that deep biology of the different tastes is connecting us to, for me, in my current journey, with the most ancient systems of medicine. So for me, that is additionally fascinating. Now, if you look at this, the technology, where the technology has gone. So we started off with sequencing a single viral genome, 5x174, etc., to where we are today. It says single cell RNA sequencing. So there is bulk sequencing and single cell sequencing, and it uses computer packages. We had the Staden package many decades ago in 1980s. Now we have the Surat package, and you can access this at this website. So this is where things have gone, and this is where that history has taken us, the history from where technology was developed to where we are today and where biology might want to go. I stop here just to point to the fact that there might be a lot of new biology, might well have rules that connect with very, very ancient knowledge from 2,500 or 5,000 or more years ago. And to access all of that, we're going to need new technology, and we're going to have to find new ways. But all of that will depend upon using the technology that is based on harvesting and being able to analyze large amounts of data. And that the large amounts of data would relate to data that might come from even very small samples. So I call this big data from small samples, sequencing a single cell, identifying. So it's big data from small sample. So everybody talks about big data. And I just want to push the boundary by saying big data from small samples and big data from dynamic samples. That's another thing that we will touch on. So dynamic samples, and the one I'll come to as we go along, is to do with the microbiome, an extremely dynamic sample. So complex data, very dynamic because it changes every day, it changes as you get older, and changes depending on what you eat, and they have a huge impact on your health and state of health and disease and progression of disease. And it involves even more complicated analysis where we have to understand the metabolics of these, of what happens from the microbiome, etc., etc. So in all of these things, the first point I want to leave you with is that you need to have some understanding of, number one, the problem and the problem space. But more importantly, it's extremely important that you have some understanding of the technology that is used for handling huge amounts of data. So, having said that, I want to now go into just sharing a few web pages. It's accessible to anybody, but based on my understanding and my background and my history and my wishes for the future, I will take you through a few web pages and show you how the whole area of data management and data engineering and data information management all becomes a central part of biology today, and is going to be a more important part as biology progresses and as we go into the future of biology. So once again, I share my screen. While they tell me if this appears, if it's on the screen.
I'm yet to see your screen.
The point I want to.
Okay. Yeah.
Is it up?
It's up.
It's up. Thank you. So the last on my PowerPoint, that's where I got the details from. That's that paper, and that paper has all the data. It's useful to study to see how complex and the kind of teamwork that's involved in putting such information together. Is my screen visible? It is. Thank you. Okay, so where do we begin? I'm going to start perhaps somewhere here. Okay, my first point protocol today is the Human Genome Browser, the Genome database. And this is running out of the University of California in Santa Cruz. Now, let me go back here and see if I can trace this link back. I want to go to the opening page of the website, and then I would like to bring you back to the ACE2 receptor. And the way to do this would be to, in all of this, I want to keep, the point I want to convey is I thought it'd be a good idea to do this on the fly and access websites and see how all that information that's available to everybody can be accessed from any part of the world. You can do it from wherever you are. There is a problem connecting to this. Let us see if we can get a mirror here. And, if that doesn't work, it doesn't matter. We can find another way to do this. So I'd like you to note this website here, the University of California, Santa Cruz Genome Browser. And if you go in there, the website might come up at some point, and we will be able to access and find a way to go into different parts of that website, look at how complex data sets from different databases are organized and how you can access all of those things from different parts of, from any part of the web page. You can see expression of this protein, the ACE2 protein. Now, for those who are familiar with the ongoing emphasis on ACE2, what is exciting is to look at how and where the gene is expressed in human tissues. And you can look at the expression here. You can see it's expressed predominantly in the small intestine. A little bit, second is in the testes. There is a little bit of expression here in the kidney. There's a little bit of expression here in the left ventricle of the heart, and so on. You'll see in colon, there's a little bit of expression in the transverse colon. See how specific one can be. One can see expression here in visceral adipose tissue. Now, for those, I think everybody knows ACE2 because it's been so much on the news about being the receptor for the coronavirus that's causing problems to all of us around the world. And this is the power of information. When information is gathered, harvested, and curated, it gives us a very good idea of where biology plays out in terms of pathology, in terms of how we can go about curing things, perhaps. So this is the power of data, and that data has been gathered very carefully and with a lot of pain, and then reorganized, and then it's got to be displayed so that it becomes user-friendly. Most of us wouldn't even know how to do this, but there are people working in the background who are experts at organizing data, harvesting data, and displaying it so beautifully here for those who are not interested in that space, to the space of data gathering and data management. But as an end user, we can start to use that information. It seems like there is a problem with accessing UCSC, University of California. So we'll come back to this. Maybe the server is down or there's a problem there, but we'll come back to this. We'll come back at the end, and I'll take you through that journey then. Now, the second thing that I just briefly talked about, as I mentioned, about dynamic genomes and how to study genomes. And this is even more challenging because the genome is extremely, the complexity of this metagenome is beyond belief. The number of bacteria that are there and other organisms, bacteriophage that are there, the nature of how you collect the sample and how you may get incorrect data if something is not correct, how you have to then prepare the DNA to get it ready for sequence analysis, and how you then harvest that data and display. So in here are some of the studies at this web page. Would you like me to put this website on the chat? Maybe I'll just put that in there so that those who are interested, they can access this while. Okay, so it's just a quick run through. See, look at this. So they're looking at the microbiome of the bee, insect microbiome. A Bangladesh 5-year-old study is another big study. Mosquitoes, microbiome of mosquitoes. And then if you go into diabetes and the immune system, there's some work. Even more exciting, see this daily baby study, near daily fecal samples collected from term infants during the first year of life. Now, can you just imagine the complexity of this study? So on one hand, the technical challenges of that study, that you have to collect the fecal matter, and then you have to do the analysis. So as you go further in, you will see a family microbiome study. This is to unearth the genetics of microbial genome control. You can see the amount of data that's here. I think there is, let me just see, there is one here which I thought would also connect us with Africa, because you guys see this study from Uganda. It's a study profile microbes present in the cerebrospinal fluid of pediatric patients with infectious and non-infectious hydrocephalus. So 92 children from Uganda taken and their microbiome study. So this is gives you a rough idea. Now here's another study from Uganda maternal taxon. And see a very quick idea. You can get a good idea of the complexity of the data and the kind of number of bacteria that they're analyzing, the number of families that they're analyzing. On the left-hand side, I hope my thing is scrolling. Look at that. That's the complexity of this data. Hundreds of genomes to be analyzed. And you then have to arrange the data, you have to present it correctly, etc., etc. So that was one example. You just go back and a quick run through the projects that are going on here, and you can see the range of projects and the kind of challenges that will be there for both the experimenter and the one who's managing the data. Right. Website here. Sorry, this is not quite in sequence, but I just want to bring the historical context that I talked about. And this is the historic Medical Research Council Laboratory of Molecular Biology here. Put that in the chat box for you. And this is where Roger Staden worked. And if you look at the historic timeline of this lab, then this is what we see. The LMB stands for the Lab of Molecular Biology for the Medical Research Council, and the historic timeline, 1947, when the lab was established, and 6 years later, in 53, the double helix structure of DNA was elucidated, and 58, when Fred Sanger won his first Nobel Prize, and then they go on to look at the protein structure, etc., etc. And the first Fred Sanger wins, you see in 1980, he wins the second Nobel Prize, etc. I won't go into the details, but you can see how one single lab has given rise to almost all the major discoveries. This lab, I think the current count is 16 Nobel Prize winners in this lab. And how a single lab has worked beyond the standard, the usual barriers between disciplines, and how they've managed to enable new ways of thinking. Link on the chat box, so for those who are interested, you can access that and have fun. In terms of the story of one person, I highlighted this lovely person here, her name, she's the center, the Genome Center here in Cambridge, in the village of Hingston. And Georgia, as you can see, I'm just going to read that put out. Georgia is a data scientist working in the parasites and microbes group. Her interests surround using computational tools and genomic data to improve. They have to do a little bit of editing here. You can see a bit of duplication, improve human health by furthering knowledge of disease. So I'm going to put that in the chat box so that you have access to this. And the reason why I highlight this is because I want to present, it seems to touch very, very nicely in the context of the aim of this of today's meeting, which is to find out how a person has come to be where they are. And academic history is very recent. In 2017, she commenced undergraduate studies at the University of East Anglia and awarded a certificate in medical science. And then in 2019, she started her undergraduate thesis at Earlham Institute, which is a very valuable institute. So please search that website just to get an idea of where this institute is. And then she graduated in 2020 with a first-class honors from the University of East Anglia and went on to find a job. And hopefully staying in this position, she will go on to get a PhD if she's interested. So you have examples of this kind, and you can use that to get a rough idea of I don't know how we are for time. I think I would like to stop here. I've shown you very quickly. I have shown you where the historic context. I have taken you through a very quick run through a few of the websites and where things are. And I've taken you into the story of one person who's currently working at the Hingston Genome Campus. And you can see from there, I put the links for all of these on the chat box for those who want to access and have a look through. So Tolani, with your permission, I'm going to stop my part of the story. But do permit me to just quickly see if I can access that Genome Browser webpage because that has a little more detail on certain genes and certain things that I wanted to touch on. If we'll come back to this. Maybe we'll give it a little more time, see if the server comes on, but otherwise you know where it is. I'm going to just put that the link for that on the chat box so that it stays here and we can come back to it. Bear with me, I'll just pull that website, Genome Browser. Okay. There is a mirror for this here in Europe, but I don't seem to be able to exit that mirror either. So Tolani, there we are. I'm going to put that, the Genome Browser web page, link to the web page here, and then we will come back to it maybe after after whatever we wanted to do now. So I hope I've given you a brief taste of how it is becoming even more important to be familiar with how the volume of data that is being used in biology today and how it has become so specialized. There are so many other resources that is perhaps, okay, let me do, no, there are so many other resources, so many other projects. That is, the beautiful thing is that it is accessible to any of you from any corner of the world. Doesn't matter where you are, you have access to, and you can reach out to those places. You can find out who is working in these places, what are the projects they're working on, and start dialoguing with them. And that's the first step to finding your next, the next stage of your career. I'm assuming many of the people who have gathered here, it's so wonderful to see almost 50 participants here. That includes me, of course, who joined us today. So thanks to all of you who are here with us today. Thank you for joining. Thank you for expressing your interest in this presentation and this discussion. And thank you for joining us. Now I leave it to Tolan and Michael and Wale to tell us where we go. What I discussed with Michael is that we will now open the floor to discussion, question answers from whoever is there, and see where the discussion goes. So thanks from me. I've enjoyed doing this a little bit. The only glitch I saw was accessing the University of California. So thank you.
Thanks, sir. We enjoyed it. Thank you very much. Yes, and that question is there a specific kind of educational background that is suitable for bioinformatics?
Is there a specific background? Yes, it's called curiosity. That's the background. So okay. You have to be curious. And yes, Do you see what I mean? You must be curious. You must be curious to find out more. When I came to, preparing to come to Cambridge in 1979, 1980, I didn't have access to any of these things. The only thing access I had to was printed journals. And we had to sit down in the library, we had to look through the journals, read the papers, and then identify people and then get the postal address. There was no email then. You had to get a postal address. You had to write a letter, and mostly a handwritten letter, and wait for a few weeks to get a reply, if they cared to reply. Most times they did. So that whole process remains, which means, number one, you must be curious. Number two, you must go through the process of identifying those labs, identifying those research areas, something that resonates with you that you like, and then connect with them. Life is made much easier because you can access, you can see them sitting in the comfort of your home. You can see where they are. You can get their email address. You can connect them, connect by email. So that is the, I feel that is the background. So that is that is what you need to do. And the rest of it, I'm assuming you will be taught in your courses. So if you are in an undergraduate program or in a master's program on information management or bioinformatics, then all the background details will be provided, but that is only part of the story. The real part of the story is to take that knowledge that you gain and see whether you resonate with it, how you resonate with that. You know, what is it that you like? What is it that you like? And how do you go about, what do you want to do next? I was crazy about DNA, and I was only 16 when I wanted to do DNA. And that craziness stays with me even today. Even today, I'm crazy about DNA. I don't care about proteins. I don't care about RNA. I'm fascinated by DNA. So what is that something that will make you crazy enough to do crazy things? So that is my answer to the question. Now I can see a question here saying, from Bolu Watifa, saying, do you have any suggested program for people who want to do grant studies in the intersection of wet and dry lab? What is your experience and advice with this kind of work? Is it advisable? Certainly, that is the way ahead. You have to be with both those skills. You have to understand lab work. You have to understand how to generate high quality data. And if you can do that as part of your PhD, along with handling that data and learning new skills, then you get extra marks for being able to do that. So you're bringing together many skills, the skills of working diligently in the lab, and learning how to harvest data from complex experiments, and then how to manage that data, how to mine that data, how to make sense of it. If that website, the University of California, Santa Cruz website comes on alive, I will take you into one example about telomere biology, and I just want to leave you with a question there. Let's see if it comes through. So thanks for that question. Again, I'd love to show you one or two examples of where you see this intersection between life science and wet science. I'm going to Abraham. Abraham, you raised your hand. unmute and let's hear you directly from you. Abraham, can you hear me? Abraham, you have
Hello,
to
brother. Can you hear me?
Abraham, I can hear you clearly. Thank you for joining us today. Thank you for your question. What's your question?
Thank you so much for having us so far.
Now, before I let you ask your question, let me ask a few questions. Where are you joining from today?
Okay, I'm joining from Nigeria today. Abuja.
Where in Nigeria are you?
I'm
Lagos?
joining from capital Abuja. No, Abuja.
Abuja. Welcome. Thank you for joining.
Okay, thank you so much for having me. So I'm just going to give a brief background about myself, then I'll go ahead and ask my question.
Abraham,
Okay.
do you have bandwidth enough to turn your camera on so that we can all see you too?
Okay, I have bandwidth enough to turn on my camera. It's just I just hope I can. The background noise here
I think
doesn't. If you
Tolani
can
might
hear
have
me
to
well.
give you permission to turn your camera on.
Okay.
But if you have bandwidth, then I think we'd love to see. Oh, there
So
you
maybe
are.
see my
Hello.
camera now.
Hello.
Hello. Yeah, hello. A brief review about myself. I graduated in 2019 and I studied biochemistry. So since then, now I'm currently a machine learning engineer. I believe you know the field of machine learning and artificial intelligence. So I've worked a while and I've garnered experience in that field. So now I'm considering graduate school, and I'm looking for an intersection between biochemistry and artificial intelligence. So bioinformatics seems to be like the best fit for me right now. Right. So, but when I check some graduate school curriculum, because I'm really particular about the curriculum, what I want to learn in graduate school. So if I check a course, I go check the curriculum, and most of what I see are biologically related things like really biology, and you see very little about writing codes and trying to build things in the technological space, which is what I do currently, and I enjoy doing that most than the biological and lab work. I enjoy doing the coding aspect of things. So I'm trying to see how can I balance this, considering that most of the courses I see in bioinformatics are like lab works and more of pure biology than the technical aspect of it. So that has been my challenge now, for a while, and I don't know if you could offer me some suggestions. Thank you so much.
Thank you, Abraham. Now, you are expressed your wish very nicely. Thank you for sharing that. And it is not a rarity to do those two things, you know, to want to excel in lab work and also excel in coding. For a lot of people, this might sound strange, but it's not strange at all. So number one, what you've expressed is something that is feasible. Now, you clearly have said the other thing, which is to enable this, it will be good if you found your way to a lab or an institute or in some part of the world, which will encourage you to do that. So that's very important. You want to specify, like you've specified now, you want to specify this to the people you are interacting with. And you want to continue excelling in lab work, bench work, and you also want to do coding. You want to write new things. Now, what will happen is as you go into those two spaces, you will find that the time required for both those will limit you in how much you can spend time in one side or the other. And this is generally what happens. There are only 24 hours in a day. And some of the lab work, wet lab work, can be very, very demanding. And the same thing with coding. If you will find there are a lot of libraries already available. You know, people have written code that is already there, and some of the projects there might be very, very challenging to work with. and they form big teams who then take on, I think there are even large companies that are coding, and to make that code meaningful and to present the analysis correctly, you need very large teams of people. So number one, find a place where you can do those two things together and see if you can identify a lab, a principal investigator who's doing that, or at least you're in an environment where you can do both those things. It might not be in the same lab, but it could be in the same city. It could be in the same institute where the tasks are divided, but the people are all there together. So that's my answer. And sitting in Cambridge, I would suggest, Abraham, to look at Cambridge as a place because it's a small city. It will give you the canvas to work on whatever you have in your mind. And it will also challenge you if you feel that is what you need, a challenge. Then you have more than enough challenges in the small city. City is so small, you can walk around the whole city. And from what you've seen, what I've shown you, you can see there are so many innovations that have happened in the city. So, Abraham, my answer to your question, try and look at a place like Cambridge. Maybe there are good labs in America, there are good institutes in America. If that's where you want to go, identify those labs. I'm going to just see if I can locate one place where such work is going on. And the gentleman, the lab is one, his name is Madan Babu. Once again, let me see if I can share my screen so that you know what I'm doing. And you, Talani, I'm just going to share this screen. Tell me if it appears here. Is the screen on? Can you see my screen? So, do you see my, Abraham, can you see what I'm searching through? And it will take you to this is just one example. This is a gentleman named Madan Babu, and Madan is let me just put this in the chat box. Oh, so here is one area where you will be able to do both those things, Abraham. I'll put that in the chat box here. And he's very keen on all of these things. And these are all areas where you will have an opportunity to link coding, new code to solve new problems, and managing data, etc., etc. So I'll put the link on the chat box. And this is just one example. There are many other places, many other locations where you can where you'll be able to access the combination of the two things that you want. Hope that's useful, Abraham. I can see you.
Yeah.
Thank you.
Yeah.
Okay, now.
I think we still have a few more questions.
Hey, how are you doing?
Yeah, I'm fine.
Okay.
We still have
a few more questions.
Yeah.
So Abraham is a happy customer.
Yeah,
so he's
you were able
going
to...
back. He's a happy customer. Okay, thanks, Abraham. Happy we could satisfy you. Now I'm going to work on that. Work on the questions. Talani, I'm just working through the chat from the bottom, from the last one. I've started.
maybe I should read them for you so that you can, and because
No, it's good. I've started, this is from Kudaisi. I've started learning Excel, data science, Python, and of course, Coursera on Bio. What are your advice for me to leverage it to get, could you permit Kudaisi to chat, to use the microphone? And I can see Jay Kishan Solanki saying, can you guide me with the things essential to get fellowship in PhD? Jay Kishan, let's come back to your question, but let me just address Kudaisi Raifu.
I'm not sure I can find I can't find him.
Kudaisi, could you unmute if you know where you are?
Not everybody can unmute, but if you can raise his
everybody.
hand, yeah, if you can raise his hand.
Raise your, Kudaisi, raise your hand, and then we'll be able to see where you are. And the second one, there's another question from somebody who's going with a screen name, Juxi. Oh, there is Kudaisi. Hi.
Okay.
Kudaisi. Yeah, thank you.
Just, I've told him to unmute himself, so.
Okay, unmute Kudaisi. And you can.
Hello.
Hey, Kudaisi, where are you joining us from today? Thank you for your question.
Hello, sir.
Hello, can you hear us? Hello. We can hear you.
Yeah.
Kudaisi, we can hear you. Keep talking.
I can hear you. Can you hear me too, sir?
Hi. Keep
I said,
your question.
I'm a pharmacist. I had my first degree is in pharmacy, and I wish to go for genomic medicine to drug discovery for my MSc program. So I just need advice and guidance on what to leverage on. I started learning. So I started in Excel, Python, because I had that I need to learn how to code.
Kudaisi, we have a problem.
So I started a course on bioinformatics. Yeah.
There's a bandwidth problem. Can you hear you clearly? My apologies.
Yeah. Okay, I think this is okay.
I heard what he said, so let me just.
Okay, please tell anything.
So he said he's a pharmacist. He studied pharmacy, and he wants to go for his master's in genomic medicine and drug discovery. So he wants to know what he can do to like get into drug discovery and genomic medicine. He said learning something.
Thank you, Telani. Thank you. You know, there's a resource, a web resource called Find a Masters. And if you just Google find, just one word, Find a Masters, it's one, right, without spaces, Find a Masters. Tolani, can you see that web page? It's called Find a Masters. And there is also a website called Find a PhD. Yeah, Find a Masters. I think it's findamasters.com, I think, but just Google.
I'm going to send
it. Find a there it is, findamasters.com.
Okay.
Michael, thank you for that. Yeah, Michael has just put that there. Michael, could you also put findaphd.com? I think that's called findaphd.com. So my comment in reply to that question from Kudaisi is just look through that. Just go in, and it's got a lovely, easy to use search box, and type in whatever you want, and you'll be surprised what's there. Just type in whatever you want to do, drug discovery, or you want to do, and you will find all that information there from around the world. And so that's my advice. What do you think? Is that helpful? Have a play with that. And then if it doesn't work, then get in touch with Michael or Talani, and then we can see how to address your question.
Thank you very
How
much.
does that sound, Talani? Does it sound all right?
Yes, it definitely sounds all right. Find
A little bit of work, your side too, just to spend some time just looking through and access. Now, where was Tolani? Where was could I see from? Was he from? He said Nigeria, but where in Nigeria? Was he joining from Lagos or from Abuja or from?
He didn't really introduce himself.
He didn't give the name. Okay, sorry.
I think he's in Nigeria.
Okay.
Yeah, it sounds like a Nigerian name, right?
Yeah, it's a Nigerian name, but I don't know where exactly.
Where you don't. Okay. Well, I'm sure we've given some homework and then come back so that we can discuss this further. Now there's another raised hand. Titi Lola. Titi Lola, hello. Titi Lola, where are you? Thank you for raising your hand. We unmute.
Hello.
I think Mike. Hi, Titi Lola. Unmute, and then.
Yeah, look, can you hear me?
I can hear you clearly.
Okay, I'm Titula from Lagos, Nigeria.
Oh, wonderful. Thank you for joining us, and thank you for raising your hand. Wait to your question.
Okay, sir. So I'm a graduate of biochemistry.
Now, Titi Lola, do you have bandwidth enough to put your camera on, or will that slow your audio?
No, I'm actually on the road, so I don't think I have turned on my camera. Yes.
Well, I was hoping we could also see what's in the roadside, but there it is. If you don't want to show us what's happening there, then that's fine. We'll take your question.
It's maybe a distraction, that's why.
That's fine.
Okay, so I'm a graduate of biochemistry, but my BSc thesis was based on COVID-19 virus and like drug discovery in terms of targeting. So with that, I got interested in bioinformatics. But right now, I'm seeing that I need to have like a knowledge of Python or R. Well, I don't know whether it is best for me to learn the two programming languages together or if I can just pick one since my focus is based on drug discovery.
Thank you, Titlola. Your question is wonderful. Now, how far are you into your studies? I didn't get the first part. Are you a master's student or just completed your?
I just finished my first degree.
First degree, well done.
Yes.
Now, your question is important. Now, what I suggest, which is for many other people too, is, of course, keep your interest higher in coding and learning a language, etc., etc. But more importantly, find a lab, find a location where you can go and spend some time. What happens here in the UK is very interesting. We have just close to my house here is the high school. And that high school has a research lab. And it is located just next to the lab of molecular biology and the Cancer Institute and the other institutes. And there are high school children who get a chance to engage in projects. So you can see the advantage they have. So by the time they have finished high school, they have been in a lab and the world's best lab. And they've learned how to, they are not worried about pipetting and they're not worried about setting up an experiment. You can just tell them, do this experiment. They're only in high school and they'll do an experiment for you. Now you see the advantage and they have and the disadvantage we have. And we got to find a way to level this, you know. So what I suggest is that you find a lab where we can go and spend time, either in Nigeria or in Ghana or somewhere, if not in Africa, maybe in elsewhere. Is that something, Titilola, is that something you are able to do? Is that something that you're excited about, or is it too frightening, or is it something that you can, you know, say, okay, this sounds exciting, I'm ready to go now? And I'd suggest something like that. And then when you go to, it's one thing learning these things. Yes, you've shown you're interested in coding and you wanted to go in a certain direction. It's another thing to find your way into a lab and to go and spend maybe 1 month in a lab, working with people who are really working at that cutting edge area, and gain some lab experience. And then you really see what this is all about. So that's what I feel you should do. It might seem like a long shot from where you are, but I think if you don't try, you don't know. So that's what I would suggest. And there are places where you can get funding for undergraduate projects here in Europe. And I suggest you should try and find a place like that. The second thing is to see if you can get into a master's program. And a master's program is a little more structured, and you are taught certain things, and you get to know all of these different programming environments and languages and problems that people are solving. And then you get a chance to work on a project. And that's a very comprehensive way to do this. So we can discuss about Titilola. I see you're back on the thing. Maybe you have a question, but could we we're going to hold your question for a while we take, how does that sound, Michael? We go to Jay Kishan.
Okay.
Titilola, don't go away because you are unmuted, but we'll take Jay Kishan's. Maybe there is something there from Jay Kishan's question that will add to what you have asked. Jay Kishan,
Yes,
namaskar, kahan se bol rahe ho?
sir, I am from India. I'm studying, doing my master's in bioinformatics from Central University of Pondicherry, India.
Very nice.
Sir, I have been there in bioinformatics since five years. I have done my UG and master's also in bioinformatics.
Jay Kishan, I'm going to stop you here very quickly. Before you go off the line, could you put the link to your university here? Because I think this resonates a little bit with what Titilola was asking. So for everybody's sake, please put the link to your institute in Pondicherry and the course. If it is available, then please put that. Please continue with your question.
Yes, sir, I will put it. And sir, my question is that it's the peak time that I'm just going to complete my master's, and after that I'm looking for the PhD position. So I want to know that what kind of skills or what kind of things I should have to get a fellowship in PhD.
Thank you.
In bioinformatics only.
Thank you, Jay Kishan. Number one, are you doing a project in your master's, for your master's thesis? Is there a thesis project? Jay Kishan?
Yes, sir. I did a project in my master's, as well as I do a few project, one project in my UG. After that, two other project from outside, and now currently I'm doing one project, research thesis.
Okay. Now, what I suggest, looks like you have already a lot of experience in doing projects, and maybe you want to share that also on the chat box of the projects you have completed, and when you're pointing us to the website. What was the most exciting project you found out of all the things from your undergraduate to your master's? And that is important, just so that you can have a feeling for something that you've done. What is it that you found most exciting out of all the projects you've done? So that's number one. Number two, you can phrase that while I'm talking and tell us all what you've done. Number two, have you prepared a good resume of all the things that you've done? And number three is, have you prepared a good, convincing covering letter? Your statement of purpose, a good covering letter, and a resume. So these are important things. And the other thing is to start looking at the Find a PhD, that website, findaphd.com. Just look through that to see if there is something that resonates with your interest. How does that sound? So in there, you will find a lot of positions that are advertised that have funding. In some countries, they prefer students from those countries. I'm told there is, Finland is one country that has an opportunity for people from other parts of the world. They like to bring people from other parts of the world to Finland, and often they also have fellowships for students. Keep in mind, it's a very competitive area where people want to have students from their own countries. So that is my answer, Jason, to your question, to your query. Look through findaphd.com and see what is available there and what is going on. In the meanwhile, please give us phytochemicals for disease, particularly cancer, to NGS, how gene expression changes. Same. Thank you for sharing this. Please, Jekishan, please also put the website. Okay. icpu.edu.in. Wonderful. I think this is going to be very, very important. I won't look at it now because we are little, we have a few more questions to address.
Yeah, I think one other thing to take note of is that we still have another session.
Okay.
So, yeah, where it will be a little bit more interactive.
Okay.
Maybe there will be no presentations and everybody will just come with questions.
Questions, okay. Okay, so what I think we have another session planned for next month, am I right? Where it'll be a more interactive session. And I think there will be an opportunity. But I want to take
let's take
Tolani, I just want to take this one lovely long question here from Shola.
Okay.
Emiade. Shola, raise your hand so that we can also, I'll read your question. If you raise your hand, then somebody will let you in the room. Thanks for the insights you provide so far. I have a first degree in biochemistry and a master's degree in pharmacology. I'm presently doing a PhD in pharmacometrics. I have a keen interest in developing gene therapy and DNA vaccine. I'm also bioinformatics. Okay, wonderful question. There is Shola. I think if you unmute it, then you can.
Yeah, I told him to, I'm not sure if it's him or her.
Shola, hi, Shola. If you unmute, then you can talk us directly. And there is, please can you address the question I sent it previously, sir? I'm inquiring from Damilola. Damilola, where is your question?
Hello, good evening.
Hi, who's that? Shola. Shola, hi. How are you? Where are you?
I'm Shola.
Where are you talking today?
Yes, I'm joining from Kaduna State, Nigeria.
Wonderful.
Northern Nigeria.
Wonderful.
Yes. Thank you so much for this opportunity and the insights you have provided so far. It's been a very wonderful session for me.
Thank you.
Yes, sir. Like I typed in the chat box, I have a first degree in biochemistry and a master's degree in pharmacology. And I'm seeking for a PhD opportunity in pharmacometrics.
Where is this,
but
in Nigeria?
Lately no, abroad.
Abroad, okay.
UK or US probably. Yes, although I narrowly missed an opportunity. I got informed lately in Denmark, a scholarship opportunity in Denmark. I missed that narrowly. So lately I got quite interested in bioinformatics too, since both bioinformatics and pharmacometrics both involve coding and the rest of them, I am actually interested in that too. So the focus of research I am particularly interested in is gene therapy development and DNA vaccine development. So I would really like to know if there's an intersection point between these two fields. I'm interested in both of them, and I want to see how I can leverage on knowledge on both of them to be able to achieve the goal of developing DNA vaccine and gene therapy. Thank you, sir.
Thank you for the question, and thank you for pointing us to this fascinating. It's the first time I'm hearing about this area called pharmacometrics, and very happy to learn a new term. Thank you for pointing me and pointing the rest of us to this new term. I feel with all these things, the question of intersections, there are intersections at all points, and it's left to you to visualize, as Tolani, when she introduced me, she said, painted the kind of things I have done. I started, I wanted to study medicine, but I ended up studying agriculture. Then I specialized in plant breeding and genetics. Then I did my master's, and then I worked and came to do a PhD in molecular genetics. And it's gone on in that way. And wherever I see, I can only see intersections, number one. And let me give you a rough idea of the kind of canvas I would use to see these intersections. The canvas I use, for instance, is, which everybody is talking about, is the One Earth, One Health campus. Okay, so that's the canvas I'm using. So if you go into that space, you will see intersections of absolutely crazy things. You will see food and health, just food and health. You will see intersections between microbial genomics and cancer genomics, for instance. You will see very interesting intersections there. So it's left to you to keep your mind open to see the big canvas and the flow of knowledge within that big canvas. So take simple thing, you might, if you go into the whole space of One Health, where we look at environment, humans, animals, plants, all together. So don't go too fine into smaller areas. Look at it in its entirety, for the moment. Just see how you can look at a very wide canvas. And in that wide canvas, you will see threads. So I showed you, on the microbial, the microbiome genome space, we looked at the microbiome of the honeybee. Now, isn't that fascinating? Because in there could be surprises. There could be some amazing surprises that will help us with understanding, because it's such a sheltered ecosystem. There might well be surprises that will lead us into other areas of relevance to, say, maybe finding a new way to manage pests in plants. Who knows? So keep an open mind on one hand, but at the same time, narrow yourself in to find problems. So this is the way I approach this. And this is my message to all of you here. Don't focus too much onto something right now. You're too young to do that. Keep your mind open to all kinds of possibilities where you can interact and learn new things. So it shouldn't be just learning Python. It could be just keeping your mind open for whatever comes your way, whatever opportunity comes your way. Yes, you might want to say, I'm interested in proteins or RNA or long non-coding RNA or whatever it is, or small microRNAs, or it could be any of those things, but keep your mind open. And that is my suggestion at this point. And use those resources effectively. There are lots of resources online, and try and use those resources. I see a message here from Helen Dennis. It says, I have BSc in Biomedical Sciences and MSc in Health Information Technology. I'm looking for a PhD opportunity My interest is in genomic science and proteins. And Helen, are you coming from Uganda? I think is that address. Can Helen Tolani, could you give Helen an opportunity to ask her question directly?
I guess I cannot find Helen here.
Helen, raise your hand and then we'll be able to see you somewhere here. It says Helen Dennis. So Helen comes to us from the Mabarara University of Science and Technology. This is wonderful. I think we would love to. Helen, are you there? We would love to hear from you directly, Helen.
Yeah, I told her to unmute.
Okay, I can't unmute myself. Okay, you have to permit her.
I've told her severally. So yeah, there's a. Hopefully she can. Yeah, she's on.
Yes, I can. Thank you very much.
Helen, hello. How are you?
Oh, good evening, all.
Good evening. Lovely to have you here. Are you joining us from Uganda today?
No, I'm back to Nigeria. I'm in Nigeria now.
Okay, you're in Nigeria. Sorry. Okay, there you are.
Yeah, I just happened to have a scholarship that took me to Uganda for my master's.
Where did you do your undergraduate studies in Nigeria? And how come you went all the way to the other side of the continent? You went all the way to...
Actually, I had my first degree from Benin Republic, a French country.
Mm-hmm. And then you went all the way to Uganda for your master's.
Yeah, I got an intra-African scholarship that to do with moving away from your country to a host. Yes.
Very nice. Now, could I ask one or two questions before we address your question?
It's okay.
Now, tell us a little bit, for all gathered here, tell us a little bit about this intra-Africa scholarships. Who provides this?
Okay, it's provided by European Union.
Oh, okay.
Yes, and it has to do with specific universities in Africa.
Mm-hmm.
There are just 4 partners universities you can attend, and Nigeria is part of the partner university.
Is this the African Union?
Africa Biomedical Bioengineering Mobility.
Could you, Helen, please, before you go away today, please put if there is a link to that, I would love to have that. So, you know.
Yes, it's actually for bioengineering students.
Wonderful. Please put a link, if there is a link on where this is. But now thank you for sharing this. I'm sure there are many people listening to this who want to know a little more about this. So if you put the link there, then
okay, I will.
Then they can access that and just read. And Helen, now that you mentioned this, I'm going to put one link here for you to consider. And it's called the Gates Cambridge Scholarships. And this is not just for you, but for everybody here. But now that you've touched about, now you pointed us to what is available as intra-Africa scholarship. I'm putting in here the Gates Cambridge Scholarship. And this is this was with a scholarship that permits students to come to Cambridge to study whatever they want to study. But it's very competitive, as you can well understand, but the link is there. And look through it. It's a postgraduate scholarship at the University of Cambridge.
Thank you very much.
Did you get that? Can you see that? Now we want your question, Helen.
Okay, so based on my background, and I actually did, I recently finished an internship that has to do with designs of novel protein in Switzerland, EPFL.
Oh my God, you are a globetrotter. Amazing.
Thank you very much. So amazing. I had to, after the internship, I developed this serious interest for proteins design, because it was actually a very interesting project for me. And I found myself flowing into it. But so I was like, okay, fine. I have to go into bioinformatics because I have background in coding based on my master's study. So I'm good with Python, but I'm still learning R.
Oh, okay. No, thank you for Helen. Helen, number one, thank you for joining us today. Number two, thank you for sharing your story. I think this is your story is a model for many people who are here, that you're happy to travel around Africa from east to west, and then you hopped over to Switzerland, you did something there, and now you're getting ready for more adventures. So I think we're all very excited to hear your small story, and now additionally excited that you want to go into coding and you want to go into something something that you are passionate about. I feel, Helen.
Yes.
I think I get this strange feeling that you are happy to be challenged and you are happy to be pushed to do more things. And I would suggest you to look at those postgraduate scholarships at the University of Cambridge. What do you think? I think from what I hear, I get this strange feeling that you might just accept this challenge.
Yeah, very well. I really want something out of the ordinary, like something not common.
Now, where can we find more people like you? That's my question to you. Where are you located today, Helen? Where in Nigeria are you?
Okay, I'm in eastern part of Nigeria, Anambra State.
Okay. How do you spell that? I know the geography of Nigeria a little bit, but I need to look at this. Anyway, I'm sure you will come to us. Michael and Tolani and everybody are arranging another meeting. I think they have something else in their mind, and they will tell you when this next event is, and that event will be a very interactive event, a little more Alhambra State. Thank you, Helen. Now, my question to you once again.
Okay. I wanted to correct the Anambra.
Anambra. Okay, we got it. Okay, that's autocorrect.
Sorry, it was autocorrect.
Autocorrect, yeah, I know, it's a nightmare, autocorrect. Okay, we got it. Okay, Michael says we are running out of time. I think we should round up with Helen's question. Okay, yeah. Thank you, Michael. But I think Michael and the team at Biohack are organizing a future gathering. So if you join us, and if all who are here with us today, we had a good gathering here today, if they all join us, and maybe others too join us at the next and future events, then we can continue this discussion. I leave it there, and I don't know if I can copy this Africa. Thank you, Helen. Africa Mobility. I hope everybody is copying these websites, and Helen, thank you for sharing this.
You're welcome. Sorry, please, can I say something?
Please. Okay,
so for the mobility, they're actually out for female candidates. So it's a female candidate I can try and apply.
Wonderful. Now, I don't seem to be able to copy stuff from the chat box into my, I think there is some way to enable that, or do I have to type everything? Michael?
Yeah, it's an issue with Zoom. So what I usually do is to just click on the link directly, and then I'll go to the browser and copy it. But then I can probably share the link with you later on.
Hold on, I think I'm managing it. File. Sometimes it permits, there is some option. Okay, I got it. Thank you. What I've done is I've selected all of the text from that chat, and then it's copied over. So number one, thanks to Michael, thanks to Tolani, and thanks to all at Biohack for organizing this event. Number one, thank you so much. Thanks for everybody who's gathered here and who asked who've sat and listened to everything and asked their questions. And sorry that we couldn't access the Genome Browser webpage, but maybe we will do it the next time when we meet. Because I want to take you, especially for somebody like Helen and the other people who asked questions, I just want to show you where are the challenges where that wet lab and the coding aspects come together. And I want to take you a touch today on single cell analysis and where the future is going. And I want to take you to one or two challenging problems where you have to use the best of lab skills and use the best of coding skills to take data that's there and analyze it and to come up to reveal new biology. So we will go into all that perhaps when we meet the next time. Michael? Oh, thank you. Michael, you've been hiding somewhere. There we are. Thank you so much. I've enjoyed this. And
thank you very much, Dr. Madan. It's been a really packed evening for me, because I also got to know some new things, especially the African Mobility Program.
Oh, look at that.
You guys knew something like that existed.
Look at this. So thanks to Helen for showing us. Thanks, Helen. We appreciate, you see what happens when we start talking. Exciting things come out.
Yeah.
Helen, now I'm going to take the liberty of putting my contact details here, and this is not just for yourself, but for everybody here.
Okay.
Thank
you very much for that.
It was there on my first slide, but I will put my WhatsApp number here so that anybody who wants to contact.
Okay, on this note, we can begin to call it a day. But before we close, I can briefly introduce some of the guys that are interested in learning a few bioinformatics or data science skills. We are packing up a program for you guys. It's more of an internship where you get to learn most of the skills that you think you need, and you even work on real projects, real interesting projects. And we are collecting most of these projects from scientists all over the world, And you can be sure at the end of everything, you are really, really skilled enough, and you have something to show, For example, you're applying for a postgraduate program, you have something to show. For example, if Professor Madan is recruiting, you can show him that, I have this set of projects, I've done this set of projects, I have this set of skills, and I think I'm the best person. I think I'm the best person for your lab at the moment. Thank you very much, everyone. If you have questions, I can drop the link to our website also on the chat box for your use. Sorry, I hope I'm right. Yeah, I'm trying to put.
Okay, biohack.com, wonderful.
Yeah, so we'll be looking at, if you still have more questions, so he has shared his, Dr. Madan has shared his email address. You can direct it to him. If you have specific questions about AgBio, you can send it to him as well. I don't know if your link, if you prefer also to talk on LinkedIn, because someone said.
Yeah, Helen wants to connect on LinkedIn. Thank you, Helen. I'll as soon as I've finished this, I will. I'm not good as Helen. I'm not as good at multitasking like you are. You seem to do everything there.
It's I think it's wonderful. A demand for our generation.
Yeah, I know. So now, once again, thank you all for inviting me to your platform. And thank you for your hospitality here. I call it hospitality, and for introducing me to so many amazing people here. And thanks to all for sharing your questions. And we look forward to, I think Michael will, and Biohack will connect with all of you about future programs. And I think there may be an opportunity to meet again.
Yeah, okay. Thank you, everyone.
Has everybody copied the? There's a lot of stuff that's coming in here, so I'm just going to.
Yeah, so we have the emails, and we can always we have the emails. Okay. We have collected all the links that have been shared today, and we are going to definitely share.
Okay, with those few words, greetings from Cambridge. Thank you, and we will meet again. And you got my details, so feel free to either reach out by email or WhatsApp or everything.
Yeah, thank you guys. Thank you for such a wonderful meeting. Thank you for everyone who participated.
Thank you, Michael.
Yeah, for some reason, Tolani is a bit, I think she has to attend to a few things. So that's why I'm kind of taking charge.
People are coming up from the woodwork. Somebody named Grace. Grace, I hope you will join us in future events. I don't know where you are, but thank you for at least waving to us.
Thank you, Dr. Madan. My internet isn't so strong at this point.
Okay, Tolani, wonderful. I can understand. Thank you.
Thank you very much. I enjoyed it a lot. Thank you, sir.
Bye. Good night.
Good night, everybody.
It's very late in India, but not too late in Nigeria. Wish you a lovely evening. Wish you a lovely evening, and then we will be in touch again.
Yeah. Bye.
Good night. Bye. 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 I'm leaving now. Can I?
Yeah.
Can I leave?
Yeah.